Hey guys, so we are going to start chapter 2. In this video, we are going to go through the first two sections of chapter 2. How do we see all the little bitty guys that we started talking about in chapter 1? So here we've got a picture of two different views of organisms. We have a light microscope doing this, but you can change your light microscope and you can change the background on it depending on what you use. What we would see in our face-to-face class is this one, the bright field, which means the background is light colored and the organism itself is dark, but the field, the background, is bright. Or you can change it up, which we're going to talk about in the tail end of this chapter, and have it as a dark field where everything is dark around the organism, and the organism itself, the light is focused, the light is focused on the organism, and the organism is bright while the field is dark. So these are just two examples of different types of views of an organism, and we're going to talk about lots of them later on in the end of chapter 2. So let's just talk about the beginning. When you got to look at something, you got to use light. So we're going to talk about the properties of light. So basics, basic science stuff from grade school and high school are terms that we need to know. So light's going to come down from the sun in the electromagnetic spectrum. And when it comes down, it's going to come down in waves. And so we are going to have wavelength and amplitude. And this wave is going to have a crest up here, and then the trough is down here. And so the amplitude is going to be how tall that wave is. So I like to think about this waves, not necessarily in light. I always think about them in sound. So if you have an amplifier that you plug in your electric guitar into the amplifier, it's louder, right? The amplifier in your home entertainment system amplifies the sound and it makes it louder. So amplitude, if it's taller wave, it's going to be, it would be a louder sound than a really quiet whisper would have a smaller wave. So amplitude is the height of the peak. And then wavelength is going to be the distance between the peaks, the distance between the crests. Are they far apart? That's going to be a low frequency. If we're talking about sound, it'd be low, like a male's voice, really deep low if it's very far apart. And then a really high pitch, like soprano kind, would be a high frequency, and the wavelengths would be closer together. This also corresponds with energy levels. Low frequency levels over here are going to have a lower energy. High frequency levels are going to have a higher level of energy. And so, make sure we're getting our Bluetooth connected. So we definitely need to know wavelength and energy. There we go. Wavelength and amplitude in regards to these terms on visible light. So more terms that we need to know. So we said the light beams are coming down, right? Electromagnetic spectrum, they're coming down in waves. And we need to know about frequency is the distance between the wavelengths. And then amplitude was how tall the waves were. When that light beam comes down, it can reflect off of something. So, every morning when you're getting ready for your day and you look in the mirror, that is reflection. That is light reflecting off of a surface as reflection. Absorbance is going to be where that light energy is captured. So I like to think of absorbance as when you go out in the summertime and it's hot and you go out in the sunshine, what color t-shirt would you like to wear? Would you like to wear a black or dark gray t-shirt in the hot, hot sun? Or are you going to wear a light colored piece of clothing? Would you wear a white shirt or a black shirt if the sun is beaming down and it's 90 something degrees? You wear the white shirt, right? Because the black shirt's going to absorb all of that heat and you're going to be really hot. So absorbance is going to be where it captures the energy, and reflectance is where it reflected off. So you would want to wear the whiter shirt so it doesn't absorb the sun's energy. Transmission. Sometimes that energy just goes right on through. if you think about glass in the window, it can go right, the light waves come through the window into your apartment, into your house, and it is transmitting through that substance. So it can transmit through or it can be absorbed through. If we were shining a flashlight, beam of light, a flashlight through a glass of liquid, if that liquid in the glass was water, would it go through the other side or would it be absorbed? What if we change the liquid in the glass to Coke? What would happen with the light waves? Would it transmit all the way through or would it be absorbed? So that's something that you need to take into account. If you took majors Bio 1107, we talk about transmittance and absorption when you measure things using a mass spectrometer. You can see how much light passes through a substance, how much light is being absorbed by that substance. So absorption and transmission are terms that you will hear regarding light and how it goes through something or gets absorbed into something. Light waves can interact with each other too, so they can have interference. So with this, the textbook had mentioned ripples on water. So if you toss, your rock into the lake, then it's going to cause the ripples that disperse from where that stone went in. What if you took the rock and you skipped it and you got like 2 or 3 really good skips on it? And so it got to go pretty far. Each one of those skips causes ripples. Every time the rock hits, it's going to cause ripples, and it hits again and it's going to cause ripples. And those ripples will eventually bump into each other and then they cause disturbance. So that's going to be interference. It's interference. And then there's also diffraction, where the light waves interact with small things and they can scatter the light. So I was telling y'all about, in the previous video, about Yellowstone. We've got the Grand Prismatic Spring there. That beautiful blue color in the deepest part of it, and you'll see them in other hot springs around the area. That beautiful blue color is coming from that light coming in and hitting the particles, the sediment that is in the water column itself, that's in the water. And when it bends and scatters like that, it reflects up to us as this beautiful blue turquoise color. And it's because of the little things floating around in that water. It's diffraction. So we have covered all of these terms so far in our video. More terms. Refraction. So light is coming down and it is traveling through the air and then it hits things and interacts with stuff. So if we are comparing the light traveling through air versus the light traveling through water, like I was just talking about with the hot springs, or maybe it's going through a solid like the glass that we have here, it's going to change its speed. So light waves are going to travel faster in the air than they would going through glass here, or they would going through water. The light waves are going to slow down. And so you're going to see that light bend as it changes speed. And that's called the refractive index. And so the harder it is for the light wave to pass through, the higher that refractive index is going to be. So if we compared the refractive index for water versus air, the air refractive index is going to be very low and the water would be high because it travels faster and easier through the air than it does in the water. When it goes to the water, it slows down its speed. So the refractive index is going to be higher. Same for glass. You can see the light comes through and then it hits the glass and changes speed, and it looks like it bends when it changes speed, and that is refraction, and it is measured by a refractive index. We can see it here with this pole going down to the water. If you stick, a straw in a glass of water, you see the same exact effect. This is the refractive index, and it's because it slows down and passes through the water at a slower rate than it does in the air. Lecture quiz. So we're going to keep on going. Different lenses can change that speed. Like we saw, it's hitting the glass and then it changed from refraction. And so some of these lenses, can, if they're concave or convex, on our convex lens, we can take all that light coming through and we can focus it. The shape of the lens will focus the light beams towards a focal point, this guy right here. And so that's exactly what happens in the lens in your eyeball too. It's going to take the light that comes in towards you and it's going to focus all that light. Your lens is a convex lens that's inside your eyeball right now. And it focuses the light on this place, macula lutea, the fovea centralis in there is the place where the light is focused and concentrated. That is the focal point, that one spot where the light is going to, all the beams coalesce and go together. A concave is going to do the exact opposite, and so it can refract the light out and away from, instead of bringing it together, it does the exact opposite. So we can play around with the different types of lenses and get the light to do different things with them. So I was telling y'all, the light comes down from the sun, the electromagnetic spectrum, and so we can put it in here. And I was telling y'all wavelengths. The lower the wavelength, the further apart the crests are, lower frequencies, and then the closer the waves are, the higher the frequencies. And lower frequencies were lower energy, higher frequencies were higher energy. And so we can see it here in this picture. We have the visible light is only this section right here out of the electromagnetic spectrum. That's the stuff that we can actually see, humans can actually see. Below that, we're going to have infrared. You and I cannot see infrared. We can use infrared cameras, though, and translate that information into something that we can see. If anybody has ever watched any sort of, IR camera coming from the cops up in the helicopter way above, and they're chasing somebody, down below, and you can see that person's body heat in the infrared camera. So that's something that we can't see with a naked eye, but we can switch things around so we can see it. But infrared is going to be below our visible spectrum. Then we've, got stuff like radar and television waves. All of that is lower and lower energy levels. When we get to the visible light spectrum, we start with the red at 700 nanometers, and then 400 nanometers is going to be all the way up into the violet and indigo and our Roy G. Biv spectrum of color. So as we travel from red, which is low energy, up into the purple high energy areas. And then above that, we have ultraviolet, UV, and we cannot see UV. Humans cannot see UV, but other things can. There's lots of other animals in the world that can see UV. Insects, bees can see UV, and there's often patterns on flowers that bees see them, and it's literally like a target. And they're like, oh, that's where I need to go to get the nectar to hone in on that flower. Birds can see ultraviolet light, so they'll have ultraviolet patterns on their feathers that we can't see, but other birds can. So that's above our visible spectrum that we can see and process, but it's there and it's higher energy levels. So when you worry about sun exposure from skin cancer, that's coming from the UV waves, UVA and UVB, ultraviolet light. And then as we go up, this is even more and more concentrated energy. So now we're getting X-rays that we know, exposure to X-rays can lead to damage to the DNA and cancer. And we got gamma rays even further up. So just to give you perspective on the electromagnetic spectrum and the light coming down and those little photon energy packets, just explaining the energy levels. If you are seeing something reflecting up as white, like I was telling you about the white shirt, it's because it is reflecting all the colors back at you. When you look at behind me, I have a green screen. It's showing you the Grand Canyon from a road trip, but the color fabric is showing green to me, and it's because green is what is reflecting, but it could be absorbing other things like plants in general. When you think of plants, you think of green. They're reflecting green light back to us, but they're absorbing other colors like red and orange and blue. They absorb those guys. So some things get reflected back, some things get absorbed. Everything's different in what they do. When the light goes through, we saw that it could be refracted, that white beam could be refracted. Sometimes it gets dispersed, and so we see these super cool where you can split out all the different wavelengths, because red is a certain wavelength that we saw, and violet was a different wavelength that we saw, and we can see the different wavelengths dispersed using something like a prism. Fluorescence. With fluorescence, so we said we can't see ultraviolet light, right? Well, you and I, we cannot see this, but we can take other chemicals, other dyes, these fluorescent dyes, and they will absorb that wavelength, that ultraviolet higher energy level wavelength. They will absorb that energy at the ultraviolet level, and then they will emit it out as a different color and a lower level that you and I can process with our eyes. So with ultraviolet light, we can use a fluorescent stain. It will absorb the ultraviolet light and then send that back out as a color that we can see. So these are fluorescent dyes. They take in a higher level energy and they reflect back a lower energy level that we can see. So you can see fluorescent greens and blues and reds in this picture. And so these dyes, these are fluorescent dyes that we add to the slide, and those get absorbed. They absorb the higher energy levels, and then they reflect it back in a color that we can see. So these are fluorescents, and it's going to happen immediately, which is different than phosphorescence. Phosphorescence is going to have a delay. So the light energy is being shined onto this organism, this object, and that light energy is being absorbed. The energy is being absorbed. And then later, when that light energy is not around, it will then be emitted afterwards. So this is literally glow in the dark stuff. It's not glowing when you're looking at it during the daytime, right? But later, when you remove the light, it's glowing. So that's phosphorescence, these glow in the dark kind of pigments that we can use to absorb energy, and that they will have a delay, and then later it will phosphoresce and show this. I went on that road trip I was telling you about in Texas, and I was trying to make it. We were trying to make it to Carlsbad Caverns in New Mexico, but we couldn't make it because they closed down the caverns at 2, and there was no way we were going to make it there by 2 o'clock because we were still in Texas. But we happened to go into these caverns called the Caverns of Sonora in Sonora, Texas, and they were beautiful, amazing. I'm not into rocks, y'all, but these were these were super cool. Um, and so some of these things, they took us down the caverns, right? And it's all pitch black, and they turn on the lights so you can walk around, and there's the amazing, amazing structures. And so at the very end, they turn off the lights, and she's just got, it's pitch black. And she gives you a second so your eyes can adjust. And then our tour guide turns on the flashlight and she shines them at different minerals, different structures in the cavern. And then she's like, okay, check them out, because this one's over here, then this one's over here. And then she's like, all right, so I've shined the light on them for a few seconds. She turns off the light, and then they start to glow. So it's phosphorescent. So you see these in living things and non-living things, but this is when the object absorbs the light, and then later, after a delay, that light gets reflected back. So pretty cool stuff. You also see this happening. There's been videos of when this happens at night and the plankton in the ocean gets activated. Sometimes you'll see the waves, when the waves are curling on the crests, they'll glow at night. I may add that video in of some phosphorescence that was happening. I think it was in Miami. I may see if I can find that video and put it in the PowerPoint. But those are all super cool things where the light is being reflected back later. So we got phosphorescence. It takes a delay. There's a delay in phosphorescence. And then fluorescence is immediate. Like it's taking that higher energy level and it's sending it back out in something that you and I can see immediately. So know these two different ones. On just a few more terms that we need to know on microscopes. So we're going to go into all the different types of microscopes in this chapter. Two terms that we definitely need to know that you probably already learned when you took Anatomy and Physiology 1, if you had that, or Bio 1010 or 1107, magnification and resolution. So magnification is how big you make it. So if you're looking at a picture and you zoom in on that image or that PDF or whatever, that is magnification. You are enlarging your view of this image, of this object. Resolution, when you enlarge this image, does it still look good? Is the quality of that picture good? If you were buying a TV, right, how many pixels does it have? Is it an HD TV? What kind of HD TV? That's the resolution. So what is the fine detail on it? Can you still differentiate small details and small objects, or does it look like one big blob, or can you tell that that big blob is now two separate dots? What's the resolution? So things that affect resolution can be the numerical aperture of the lens and its ability to gather light. So the better its aperture, the better the resolution. And then another thing that can affect resolution is the wavelength. So what wavelength are you looking at? Are you using a light microscope like this guy? This is a dissecting microscope, which also uses light. Or are you using this kind of microscope, which is an electron microscope? And it's going to shoot electrons at the object, and it'll either be transmission or it will be scanning, but those are going to have shorter wavelengths, so you can see much, much better resolution. You can see smaller objects in a lot better detail. So electron microscopes can give us some really cool pictures that you will see sprinkled throughout this textbook and pretty much any other textbook that is looking at teeny tiny little objects. So wavelength and aperture can affect resolution, which is the fine details of your image. Another thing that can affect your image is contrast. If you have ever done a biology lab and you had to do the cheek cell swab, where you take the little toothpick or Q-tip and you swab the inside of your cheek, and then you take those cheek cells and you put them on your glass slide. And what's the next step if you've ever done that lab? Because if you just put your cheek cells on the slide and you stuck that slide under the microscope, what are you going to see? Everything's all clear. You have to give it some contrast. If you don't put some contrast up there, you can't tell. You can't tell what the different structures are. It all just looks the same. So you got to give it some contrast. And we will talk about all the different stains, dyes that you can use to increase contrast. And sometimes you can do it with the microscope itself, which is kind of cool. So we'll get into all those details later. But you do need contrast to be able to tell one thing from the other thing that you're looking at. Okay, so this is our case study for chapter 2. It has got to be better than Cora. So this is Cindy. She is 17 years old and she's a camp counselor. She has scraped her knee when she was playing basketball, and it was 2 weeks ago, and you'd think it'd be healed by now, but it's not. Um, so it looks like an insect bite, and it's become painful and it's swollen. So she goes to the camp nurse to have it checked out because it's been 2 weeks and it should have healed up by now, and it's nasty looking. It is oozing large amounts of pus from the wound. So the nurse is concerned because it should not be 2 weeks later still infected like that. So she thinks it's an aggressive infection. She's going to swab it and collect that sample, and she's going to clean and dress the wound, and she is going to send out the sample to a medical lab so they can see what is that in Cindy's wound. What has she got? Why is it infected? And what is the source? What's the cause of the infection? Okay, so we're going to move on to the next section. It's kind of small. Section 2.2, peering into the invisible world. So we have more names for you to know. Some of these names we have already discussed though. So this new name that we haven't discussed yet is Girolamo. I'm probably mispronouncing that. My Italian is not the best. His last name is Fracastoro. So Fracastoro was one of the first ones to think, remember we were talking about the Greek philosophers and they thought it was miasma, it was bad air. The Greek and the Romans, it was bad air. And if we can get rid of that bad air, then hopefully we can help out our situation, and less people will become ill. So it's bad air, it was not superstition. Well, Fracastoro is saying that it's got to be these tiny things. There's tiny things that are making us ill. And so he called them seeds of contagion. And so he called his book De Contagion. And he thought that these little seeds of contagion were how things were getting spread. And so they could attach onto cloth, which is called foam. And so he thought that that's how it spread from person to person, that they're little tiny seeds of contagion. And, you know, if I was infected and I am ill, I have sneezed and coughed and have gotten it on my person and on my clothing. And then maybe somehow, maybe it was my mask, right? We're wearing masks now. What if I put my mask and I coughed in it, sneezed in it, and breathed on it all day, and then I gave my mask to somebody else? That would be disgusting, right? Because my seeds of contagion are now on the cloth, which is a fomite, and I have passed it to somebody else. So Fracastoro was on to something with his book of De Contagion, and this was all the way back in 1500s. So this was Fracastoro. Other people that we have already talked about were using the beginnings of microscopes, the very primitive microscopes. So Galileo gets the credit for creating telescopes. He was the first one to invent the telescopes. He made a compound microscope similar to telescopes, right? It's got the two lenses in there. So Galileo made a compound microscope back in the 1500s. Leeuwenhoek did not use a compound microscope with two lenses. He used a simple microscope with one lens, and we already learned about Leeuwenhoek. This was like a recreation of his simple microscope. And remember, Leeuwenhoek was the first one to actually see the things in there, and he called them the wee little beasties or the animalcules. That was Leeuwenhoek. We've already talked about him. And who else is on this page? I think that's it. Fracastoro and Galileo and Leeuwenhoek. Another person that we should know about, this is later on. So 1600s was Leeuwenhoek, Galileo was 1500s. Robert Hooke was 1600s, and I think he passed away in early 1700s. But Hooke looked at slices of plants. And he published a book. It was called Micrographia. But as Hooke was looking at slices of plants and different colors of different types of plants, he had slices of cork. And so he noticed in these plant slices and the slices of cork that they had the same structures. He kept seeing the same kind of structure in everything. And so he likened it to the rooms that monks are assigned, where the monks live and sleep at night, is called a cell. And so that's what he called them because he saw them. So Robert Hooke, is his claim to fame is that he came up with cells, the name for cells, from his discovery, from looking at all of these different thin samples of plants, of cork out there, and then he published it in his book, Micrographia. So we said Galileo used the telescope. He gets credit for the telescope. What about the microscope? Who gets the credit for the microscope? So, sorry guys. It's a little sketchy. We're not quite sure exactly who gets the credit for the microscope back in the days in the 1500s, early 1600s. It could be the Jansons. And so it was a father and son, and there was Zacharias and Hans, and they were, we don't know a whole bunch about these guys, but we do know that they did mess around with microscopes. They possibly might have been the first people to create a microscope or a telescope. They played around with lenses, but we're not 100% sure if they were the first ones. One of their one of their neighbors, Hans Lippershey, may have invented it too. So we're not sure exactly when. We know around the 1500s, but we don't know, was it the Jansons? Was it father and son? and they were the ones messing around with the lenses and came up with the first microscope? Was it Hans Lippershey? We're not totally sure about that. But that does end this first video on the first half of chapter 2, and then we will talk about all the different microscopes in the next video.